Okay, I guess we'll get started. Hopefully, I know there's a bunch of people online, so hopefully you all can hear me. So before I turn over the introduction, just want to welcome everybody to this weekly installment. We're back in gear for the Pioneers in Biomedical Research Seminar Series. And a little update for next week. We have another speaker, Dr. Zheng Li will be here. Dr. Li is the chief of the section on synapse development and plasticity at the National Institute of Mental Health at NIH. And Dr. Li will be talking about mitochondria and synaptic plasticity, gamma oscillations, and anxiety. So hopefully everybody will be here for the talk next week as well. So without further ado, I'm going to turn it over to Dr. Zhishang, who's going to introduce today's speaker, Dr. Donita Brady.
Thank you, Mike. So it's my pleasure to introduce today's speaker, Dr. Donita Brady. I just learned actually last night that Dr. Brady actually was from Lynchburg, which is not far away from here. So welcome home, Dr. Brady. Dr. Brady got her Ph.D. from UNC at Chapel Hill in 2008 and did her postdoc training at Duke. I noticed that Dr. Brady's CV said that she did summer research at Dr. Shannon Deer's laboratory at UNC. You probably don't know Dr. Deer. He's a pioneer of the studying the RAS oncogene, very well-known scientist. Dr. Brady became a presidential professor and assistant professor at Department of Cancer Biology of UPenn in 2015. She's currently a Harrison Dixon and Clifford Baker presidential professor of cancer biology and associate professor at UPenn. Dr. Brady has done tremendous work in teaching, mentoring, and services, also received numerous awards. She is currently associate editor of a journal of biological chemistry and cancer research communications. If you want to submit your manuscript to those two journals, you certainly could communicate with her. Dr. Brady's work has mainly focused on copper-mediated cancer signaling. And this research is originated from her seminal work published in Nature 2014. Her recent review article published in Nature Review of Cancer summarize how copper signaling play a key role in tumor genesis. I read this article recently, and then the new concept called copperoplasia, if I pronounce it correctly, which is a copper-dependent cell growth. And so Dr. Brady's today's talk is copper-conducted kinase signaling in cancer. So without further ado, please join me. Welcome, Dr. Brady.
So thank you very much for the really kind introduction, and it's really nice to be back here in the New River Valley. So today I'm going to tell you about our work on leveraging the intersection between metal homeostasis and cancer biology through kinase signal transduction. And so normal physiology really relies on the orchestration of complex processes within cells, between cells, and amongst cells in the environment in order to regulate proper tissue homeostasis. And to really enable the precise coordination of these cellular processes with both dynamic, spatial, and temporal control, evolutionarily conserved signal transduction pathways serve as a means to both sense and respond to extracellular environments. And proteins within these signaling cascades often respond directly to these inputs. So it's not surprising that the aberrant activation of many of these signaling initiators or the inactivation of these signaling repressors is often awry in normal cells and triggers malignant transformation, where you get sustained proliferation, invasion and metastasis, and angiogenesis, which are hallmarks of tumorigenesis. And kinases within these signaling pathways has been shown to be very great drug targets for the treatment of cancer, but there are often many different reasons why these therapies aren't utilized throughout the stages of cancer. And so our lab is quite interested in protein kinases that are important in regulating post-translational modifications and many of the proteins. It's estimated that 50% of the entire proteome is regulated by post-translational modification. And therefore, the activation of protein kinases that put on these marks or the activation of phosphatases that remove these marks are quite important. When we look throughout the kinome, many of these proteins can be aberrantly activated through 3 different mechanisms. The first being point mutations, the second gene amplification, or chromosomal translocations. And the most famous of these is the BCR-ABL translocation, often referred to as the Philadelphia chromosome because it was identified at the University of Pennsylvania. And so this aberrant activation of protein kinases is quite important for that tumorigenic process. And because of that, many different pharmaceutical companies have gone after small molecule inhibitors of protein kinase activity. And it's estimated via these different mechanisms that 91 of the 500 protein kinases in the human genome are oncogenic drivers in the context of cancer, but there are only 20 FDA-approved small molecules against those 91 aberrantly activated protein kinases, and 4 are orphaned in the sense that there are no small molecule inhibitors against those protein kinases. The other issue when targeting protein kinase signaling networks is tumor cell heterogeneity and the resistance that often occurs in the context of their inhibition. One of the most famous examples of this is in the context of mutation of metastatic melanoma patients that harbor mutations in the serine threonine kinase, BRAF, where mutations at amino acid valine that convert this to glutamic acid are initiating mutations in the context of melanoma, where that mutation is found in the benign nevi of these lesions. And that led, and this discovery in 2002 led to many pharmaceutical companies looking for inhibitors against this aberrantly activated protein kinase. One of those companies, specifically Plexicon, was very successful in generating a mutant selective inhibitor, a BRAF V600E. And that was first tried in metastatic melanoma patients. Here's a 38-year-old patient who had metastatic melanoma deposits on the chest. This patient was given the BRAF inhibitor vemurafenib. And almost immediately, you could see complete resolution of these lesions. Unfortunately, about 8 weeks later, that patient represented with the lesions in the exact same place. And this was due to pathway rewiring, where you got reactivation of this canonical MAP kinase pathway. And so this really leads us to think about what are the other inputs to signal transduction pathways that may be vulnerabilities in the context of cancer biology. And so we really like to think about this in the context of micronutrients and how our diet can influence signaling pathways. And that's really born out of the idea that in organic metabolism or metabolism that we think about in terms of the complex biochemistry that's going on in most of our cells, that the metabolites that are generated by these enzymatic reactions can actually function as signaling molecules themselves. So for example, the generation of acetyl-CoA is known to be really important for protein lysine acetylation, and that can regulate the epigenome. While acetoacetate was recently discovered to be important for the interaction between oncogenic BRAF and its downstream kinases, MEK1 and 2. But unlike these organic metabolites, they can be recreated via these enzymatic reactions. Micronutrients like transition metals that are taken in through our diet can neither be created nor destroyed within biological systems. And therefore, we need strict homeostatic mechanisms to monitor metal abundance, in addition to proper dietary intake in order to regulate many of the downstream fates for these transition metals. When we think about them, they can be divided into 2 different classes. The first being redox-active transition metals like iron or copper, or redox inactive transition metals like zinc or calcium that either or static cofactors that are buried within enzyme active sites in terms of the redox active transition metals. So they're important for structure and function, while zinc and calcium are well established regulators of signal transduction through changes in the abundance of those metals in a biological system that leads to a biological output. And so what I'm going to describe to you today is this novel idea that the availability of the transition metal copper can directly regulate protein kinase activity through a direct interaction with protein kinases that leads to the increase in phosphorylation of any one of these substrates. And this new paradigm in the utilization of transition metals is something that our lab is quite interested in. And so a little bit about how copper is important in a biological system. Outside the cell, copper is found in its oxidized copper-2 state. And it's usually carried by the secreted protein ceruloplasmin. There's a ferrous oxidase that utilizes copper for that ferrous oxidase activity. Outside the cell, it's then reduced by the steep family of metalloreductases and then imported into the cell through this homotrimeric copper transporter, CTR1. Once inside cells, it's immediately bound by small molecular weight ligands like glutathione or the storage protein methylthionines. You can think about this as similar to ferritin. This is storage protein for iron. Or by chaperone proteins that are important for bringing copper to different copper-dependent enzymes based on their localization. There's a yet to be identified ligand that's important for bringing copper to the SLC25A3 protein that leads to copper import into the mitochondrial matrix, where it's then utilized by the most evolutionarily conserved copper-dependent enzyme, cytochrome C oxidase. In the cytosol, the copper chaperone CCS brings copper to the copper site in superoxide dismutase. And this is important for getting rid of reactive oxygen species, converting that to hydrogen peroxide, and then catalase converts that to molecular oxygen and water. And then finally, the cytosolic chaperone ATOX1 is important for bringing copper to the P-type ATPases, ATP7A and ATP7B, where you get metallation of many of the secreted copper-dependent enzymes like lysyl oxidase, dopamine beta hydroxylase, and tyrosinase. And then when copper levels are high in the cell, these ATPs move to the membrane and lead to copper export. So why were we interested in this, and how do we know so much about copper homeostasis? It's really based on the discovery of these rare genetic disorders that lead to differences in copper balance. The first being Menkes disease, where patients harbor a reduction in systemic copper availability because of mutations in ATP7A. So copper doesn't move from the intestine to the periphery in patients. And in this animal model, where the multibrindle animals are observed here, you see that they're much smaller in size and they're hypopigmented because there's less tyrosinase activity. This difference in animal size is not necessarily due to many of the copper-dependent enzymes that I'm showing to you here. The second disorder is Wilson disease, where patients have mutations in ATP7B, and they happen to hyperaccumulate copper in both the brain and the liver. When untreated with copper chelators, this can lead to neoplastic proliferation, especially in those animal models, and sometimes in patients where you'll get abdominal malignancies that arise. And so we know that copper is quite important for growth-like phenotypes, but many of these copper-dependent enzymes that are well characterized in the context of these diseases don't underlie these growth-like phenotypes. And so when I was a postdoctoral fellow at Duke University, we were interested in understanding the molecular nature of this copper-dependent growth phenotype. And so there I was collaborating with a graduate student, Michelle Tursky, who was in Dennis Phil's lab at the time, and they were characterizing the Drosophila homolog of CTR1. And one of the first observations that Michelle made is that if she expressed an oncogenic variant of RAS, this G14V mutation in the Drosophila eye, you get this rough eye phenotype. And that's due to the fusing of ommatidia and the overgrowth of cells. And this phenotype is exquisitely sensitive to the robustness of MAP kinase pathway activation. And so what we found is that if we simultaneously express RNAi against CTR1, you can completely ameliorate this RAS-driven phenotype, suggesting that there was a genetic connection between proper copper transport and this RAS-driven phenotype. And we knew that that was dependent upon the MAP kinase pathway. And so we then took mouse embryonic fibroblasts and asked whether or not there was a difference in MAP kinase pathway activation in the context of reduced copper transport. And what we found when we stimulated with the receptor tyrosine kinase ligand, FGF, we could see robust activation of the pathway in the wild type MEFs, but we saw reduced basal and stimulated levels of the MAP kinase pathway when we got rid of the copper transporter. Now, in work that I'm not going to go through today to tell you some of the work that we've been interested in more recently, we knew that there was only a difference in the ability of MEK to phosphorylate ERK in these cells. So all of the upstream pathway components were properly activated. And so this inability of MEK to talk to ERK led us to the hypothesis that potentially these MAP2 kinases, MEK1 and 2, may be copper binding proteins themselves. Yeah. So that's really based on the sequence and the sequence for copper binding in these protein kinases. So those same exact amino acids that we've identified to be copper binding are not found in the other protein kinases. And so that's why we think that that is selective to this particular node in the pathway. And so we tested whether or not recombinant MEK1 from bacteria could bind to copper selectively, and it did. And then in vitro kinase assays, we showed as we increased the level of copper sulfate supplemented, we could see increased phosphorylation of ERK2. And if we added the copper chelator tetrathiomolybdate, we could reduce this. And so we knew that there was this novel connection between copper transport and MAP kinase signaling strength. And so we went back to these BRAF mutation positive systems to test whether or not we could repurpose copper chelators that are utilized for Wilson disease to forestall MAP kinase pathway activation in the naive setting and the resistant setting. Because at the time that we started these studies, these BRAF inhibitors and MEK inhibitors that are now the standard of care for BRAF mutation positive patients wasn't approved. So we utilize a genetically engineered mouse model of melanoma, where we have a Cre-activatable allele of oncogenic BRAF and then loss of the tumor suppressor P10. We then enrolled mice onto 4 different treatment arms and looked at tumor growth over time. And what we found is that we could extend the time that it took to get to a tumor of a certain size if we treat it with a copper chelator TTM, and this is very similar to what we got with the BRAF inhibitor vemurafenib. And then we could extend this a little bit further if we combined the copper chelator with vemurafenib. So this would suggest us that you can combine a copper chelator in a rationally designed clinical trial with these BRAF inhibitors. More recently, we've asked, how can you increase the efficacy of these copper chelators, especially because they're cytostatic, meaning that they do not kill cells. And so we did a screen to look for sensitizers of the copper chelator TTM and found that the BH3 mimetics ABT737 could actually increase the efficacy of these copper chelators in a resistant form of BRAF mutant melanoma. And so when I started my lab at Penn, we knew that proper copper transport through CTR1 was quite important for MAP kinase pathway activation, and that this could be pharmacologically leveraged in the context of BRAF mutation positive melanoma. Well, we still had many other questions to unravel at the time. And the first was, how does copper actually modulate MEK1/2 activity at the atomic and molecular level? Meaning, what does it look like structurally? What is the coordination chemistry, and how does it enzymatically actually activate the protein kinase? Second, there's very little free copper inside cells due to Fenton chemistry that can be triggered by that redox-active metal. And so we reason that maybe one of these cytosolic copper transporters may be important for delivering copper to these dual specificity protein kinases. And so we wanted to figure out what the delivery mechanism was. And then finally, because most copper is bound to the millimolar quantities of glutathione that's inside cells, we reason that alterations in antioxidant status inside cells may be able to manipulate copper occupancy into MEK1 and 2. And then finally, in data that I'll talk about in a few minutes, we wanted to develop a specific small molecule inhibitor that was a copper chelator and a kinase inhibitor to form this novel class of MEK1/2 inhibitor. And so today I'm going to describe for you two of those areas of research in the lab. First being, how does MEK actually get its copper in a cellular system? This work was done in collaboration with my colleague, Rona Marmestein at Penn. And so one of the first experiments that Michael Grasso, who is a PhD student in chemistry, did is he took these two cytosolic copper chaperones, loaded them with copper, and then added them to MEK1/2 kinase assays and asked, can you increase the phosphorylation of ERK1 and 2 in this in vitro setting? What we found is that copper-loaded CCS could, in a dose-dependent manner, increase the phosphorylation of ERK1 and 2, suggesting that this specific copper chaperone is sufficient potentially to facilitate copper loading into the MEK1/2 kinases. And so we were interested in why there might be this discrepancy between these two cytosolic copper chaperones. And so we collaborated with Pernilla Stafid's lab at the Chalmers University in Sweden to look at the binding kinetics between MEK1 and these chaperone proteins. And we were able through these surface plasmon resonance experiments to calculate the KD of those interactions. And what we found is that the MEK1 CCS interaction is about a 2 micromolar interaction, while the interaction with ATOX1 is about 10 micromolar. And this likely underlies the inability of ATOX1 to increase MEK1 kinase activity. And so the next question that we wanted to address is, was there actually facilitated copper delivery to MEK1 through this cytosolic copper chaperone? And so Mike devised an experiment where he would load CCS purified from bacteria with copper, and then incubate that with APO MEK1 from bacteria, then separate those proteins using size exclusion chromatography, and then do ICP-MS to find out how much copper was actually bound to the MEK1 protomers. And what he found is that if he incubated equal molar amounts of copper-loaded CCS to MEK1 and then looked by ICP-MS, that he could get a 1-to-1 binding between copper and MEK1. So MEK1 has one copper binding site, and CCS is capable of facilitating loading into that copper binding site. So we were really quite excited to be able to identify this mechanism by which copper was loaded into these MAP2 kinases. And then we went back to the literature, especially from Norbert Perriman's lab, where they've done these functional RNAi screens in Drosophila S2 cells to look for regulators of the canonical MAP kinase pathway. And then if you look in the supplement to those data, you find that both the high affinity copper transporter CTR1, when knocked out, or the cytosolic copper chaperone CCS, when knocked out, can reduce MAP kinase pathway activation. Go ahead.
These also regulated by the MAP kinase pathway?
Yeah, that's a great question. So we do see some regulation of CTR1 by the MAP kinase pathway expression.
Okay.
We have not looked at whether or not it alters localization, right? Because it can be internalized, and a lot of those mechanisms of post-translational regulation of CTR1 aren't well established. Yeah, but there's a really nice recent paper in Nature Cell Biology that shows that CTR1 can be oxidized, and then it gets internalized with the VEGF receptor in endothelial cell. Exactly. Yeah, exactly. And so Both of these nodes that we had been able to identify were also found in these screening approaches, suggesting that we were on the right track. And so we next wanted to ask, do these proteins actually form a complex in cells? And so we used the BRA biotin ligase, promiscuous biotin ligase experiments to look at the association between MEK1 and these copper chaperones in cells. We needed to use this system instead of just doing straight up co-IPs because we knew that the affinity between the proteins was fairly low and they did not form a stable complex when we purified the proteins in vitro by EMSA-based assays. And so here what we're doing is we fuse the promiscuous biotin ligase, BRA, to the N-terminus of the copper chaperones. We then deplete the cells of biotin using streptavidin beads and then add back biotin for a few days, and then pull down any protein that's been biotinylated by the promiscuous biotin ligase, and then do Western blotting experiments. Nicely in the BRA ATOX1 cells, we do see biotinylation of ATP7A, and that's its cognate enzyme where we know that ATOX1 brings copper to the Golgi apparatus. We don't see biotinylation of MEK1, suggesting that they're not in close proximity to one another inside cells. However, if we look at the BRA CCS cells, we do see biotinylation of MEK1. And so these two proteins are in close proximity in biological systems. Yeah.
So those interactions are copper dependent?
Yeah, great question. So in data that I'm not going to show here, we do see some copper dependency to that interaction. So if we use a non-cell permeable copper chelator, BCS, or if we add excess copper to these cells, you can see differences in that association. Yeah, but that's a great question. We do see that using this system. And so then our next question was whether or not the endogenous CCS was required for copper-stimulated MAP kinase pathway activation. Importantly here, if we use only 1 micromolar copper chloride into the media, most of our media has very low amounts of copper supplemented. We're able to see robust activation of the MAP kinase pathway within 15 minutes, but that activation is dependent upon the upon CCS being expressed in these cells. And so we next wanted to ask whether or not CCS was required for BRAF-driven MAP kinase pathway activation. So here we're using a panel of BRAF mutation positive melanoma cell lines. We've either knocked down the endogenous CCS or CTR1, and then we're looking at MAP kinase pathway activation. And then what we find is that there is a reduction in phosphorylation of ERK1 and 2 when we knock out either CCS or CTR1 in these cells. And this correlates with a reduction in the ability of these cells to grow in these low density crystal violet based experiments. So CCS is required for BRAF-driven melanoma cell proliferation. And so is there a way to pharmacologically target the copper delivery from CCS to MEK1 and 2? We were aided in trying to do that by some work done by Siobhan He's lab at the University of Chicago, where they've developed this small molecule inhibitor of copper trafficking, specifically DCAC50, was found through some computational approaches, where they reasoned that it would block the delivery from either CCS and ATOX1 to a copper-dependent enzyme. And so this is some of the SAR that they did and modeling of the small molecule and the CCS copper binding pocket. And so Gavin Bond, who is a undergraduate, now MD Ph.D. student at Thomas Jefferson in Philadelphia, did some initial studies where he increased the concentration of DCAC50 in the context of EGF stimulated MAP kinase pathway activation. And what he found is that he could see reductions in the phosphorylation of ERK1 and 2 if he treated with increasing concentrations of DCAC50. So now we have a small molecule that's capable of preventing CCS loading into MEK1 and 2. This is similar, this reduction in MAP kinase pathway activation is similar to what we're able to see in both in BRAF mutation positive melanoma cell lines. And this correlates with the reduction in the ability of these cells to form colonies. Importantly, we only see this phenotype in the context of BRAF mutation positive melanoma cells. So if you take cells that don't rely on MAP kinase pathway activation for their growth, you don't see this phenotype.
Proliferation, or does it extend to motility?
Great question. So we didn't test whether or not it affected melanoma cell motility, at least in this assay. We know that if we, not this chaperone, but if we knock out ATOX1 in our melanoma cell lines, they do have a difference in motility.
The BRAF MEK, it can actually activate motility even in neurons that are derived.
Yeah, absolutely. Yeah, seems right. Origin. Yeah, absolutely. We think in that case, at least CCS isn't important for that phenotype, but ATOX1 is absolutely important for that phenotype because it's required for EGF to get to the cell membrane, actually. Yeah, sure. And so in summary for this part of the talk, what I've shown you is that we've been able to identify the mechanism by which copper is loaded into these MAP2 kinases, MEK1 and 2, and that there is a small molecule intervention that can be utilized to prevent copper loading into MEK1 and 2. Most of this work was done by Michael Grasso, who is a PhD student with my colleague Ronan Marmosine, and he was aided by Gavin Bond, Tiffany Zhang, and Yejin Kim, who were members of my lab. We collaborated with Catherine Alwine and Nadian Blackburn's lab at Oregon Health Sciences, Dwayne Winkler and Stephanie Boyd at UT Dallas, and Pernilla Staffheat's lab at Chalmers University to do this work. And so because of the pleiotropic effects that can happen in the context of copper chelator treatment, we know that there is polypharmacology that happens downstream of those inhibitors. We've wanted to assess whether or not we can target copper-dependent MAP kinase pathway activation selectively in tumors. And so much of this work is really inspired by some screens that were done by Liz Patton's lab, where they were utilizing zebrafish as a model of pigmentation. And what they were looking at here is they were doing chemical genetic screens to look for modifiers of pigmentation. And one of the observations that they made is that the first in class small molecule allosteric inhibitor of MEK1 and 2, UO126, can affect zebrafish pigmentation. This is not what you see when you utilize one of the Park-Davis MAP kinase inhibitors, PD03259, suggesting that this isn't a MAP kinase-driven phenotype, but instead potentially a copper-driven phenotype. Go ahead.
And this is, do you think that this is dealing with the fact that for melanin production via the tyrosine hydroxylase pathway you need copper, or is it through the neural crest melanocyte differentiation? So just so I'm clear on this before you talk.
Yeah, so we think that this is due to the copper stimulation of tyrosinase activity, not necessarily through that differentiation. Because when we use this copper chelator neocuparin, we also see this difference in pigmentation, but it isn't because there is a block in that differentiation stage. That's a great question. So it is really about tyrosinase activity in this case, and that we see an interaction between the small molecule UO126 and copper chloride in our assays, while other MAP kinase inhibitors don't have that same phenotype. And so we devised a screen to look for copper binding capacity across 120,000 compounds at Stanford University, and we were able to identify 2,100 candidates that are copper binding and then screen them against MAP kinase pathway activity in vitro. Now we've gotten down to 17 candidates that are quite selective for copper binding and MAP kinase inhibition selectivity. And I'll tell you a little bit of the data that we have on a lead compound that we call C16. And all of this work has been done in collaboration with my former postdoctoral mentor, Chris Counter, Hamid Zabrafzadi, and then a chemical biologist at Penn, George Burslem. And so first we needed to characterize the ability of this small molecule to actually bind to copper in comparison to known copper chelators. And what we found is that the small molecule C16 has about a 5 micromolar IC50 for copper binding capability. So it's on par with these typical copper chelators that are utilized in Wilson disease. And then if we look in vitro kinase assays, at its ability to inhibit constitutively active MEK1 activity, we find that IC50 is about 5 micromolar as well. So it's not as potent as cobimetinib and trametinib that are approved for the treatment of metastatic melanoma, but it does reduce MAP kinase pathway activation and could be important in the context of resistance. Importantly, it's quite selective for the MEK1 kinase domain. So if we take the N-terminus of MEK1 and then fuse it to the C-terminus or kinase lobe of MEK5 that we previously showed was not sensitive to copper chelation, we see no inhibition of its kinase activity. So it's selective to MEK1. And then when we look throughout the entire kinome, only about 20 other protein kinases can be inhibited by this small molecule. And so more recently, what we've studied is whether or not the transformation or the ability of these cells that are BRAF mutation positive to grow in soft agar can be inhibited when we treat with the small molecule C16. And so what we found is that it can reduce soft agar growth similarly to the MAP kinase inhibitor trametinib. And that if we utilize this a MAP kinase inhibitor insensitive variant of MEK1 that was recently characterized by Neil Rosen's lab, we still see sensitivity to C16 while there is resistance to the MEK1/2 inhibitor trametinib. And this is due to on-target reductions in MAP kinase pathway activation. And so what we think we've been able to uncover is this ability to utilize small molecule screening approaches to understand better how copper regulates MAP kinase pathway activation, and we can utilize this molecule to get outside of the polypharmacology that is induced by typical copper chelator treatment that induces anemia. And so for the last part of my talk, I'm going to tell you a little bit about our work on understanding copper-dependent protein kinases and screening for the copper-dependent kinome. And this is really based on our hypothesis that copper is integrated into these protein kinase signaling pathways in order to monitor copper availability to other sites like the mitochondria. So if you don't have enough copper for cytochrome C oxidase activity, you wouldn't necessarily want to activate these pro-growth like pathways. And so to get at this, we utilize a multi-tiered screening approach where we looked at the activity of 414 protein kinases in the context of a high concentration of a copper chelator. From there, we found about 132 other protein kinases that could be inhibited to a similar degree to MEK1 and 2, and then assayed the ability of those protein kinases that we had open reading frame cDNA expression contracts for, their ability to bind to a copper charged resin specifically. And so from there, we've identified 66 new protein kinases that are regulated by copper availability. And so many of these are shown here on the screen. And what I'm going to tell you about for the last part of the talk is copper regulation of the autophagic kinases ULK1 and 2 and their importance in BRAF and KRAS-driven lung adenocarcinoma. And so much of this work was inspired by, one, our multi-tiered screening approach, but also the fact that these autophagic protein kinases have similarity in their copper binding regions to MEK1. And so we previously have found that this regulatory spine residue in the HRD motif was important for binding to copper, and that these residues in the activation segments of MEK1 and 2 were important for copper binding. And we see that there's similarity between those and ULK1 and 2. So one of the first experiments that we did several years ago is ask whether or not ULK1 could bind to our copper charged resin. So here we immobilize copper, iron, or zinc, and then look for the ability of the kinase domain of ULK1 to bind to copper. And indeed, we see that there is an interaction between the protein kinase and copper. We then used our in vitro kinase assays to ask whether or not copper could increase the activity of ULK1 in a dose-dependent manner. And indeed, if we look at the phosphorylation of one of its constitutive binding partners, ATG13, we see increased phosphorylation of ATG13 as we increase the concentration of copper chloride, and that we can reduce that as we increase the concentration of the copper chelator TTM. And so indeed, ULK1 and 2 can bind to and require copper for kinase activity. And so what do these protein kinases do? They're downstream targets of mTOR when bound to the rapamycin-sensitive TORC-C1 complex. And when nutrients are plentiful, TOR in this TORC-C1 complex phosphorylates ONC1 and 2 and leads to their inactivation. However, when glucose, ATP, or amino acids are in low abundance, you see mTOR is inactivated, comes off of the lysosome surface. This leads to the de-repression of this phosphorylation and the activation of ONC1 and 2, where it can go on to phosphorylate its constitutive binding partners, ATG13 and FIP200, which then lead to the activation of the class 3 PI3 kinase complex, or VPS34, which is really important for phosphorylating PI and converting that to PI3P on this growing autophagosome. This then leads to the recruitment of 5-domain containing proteins that are then important for bringing the ubiquitin-like conjugation system that's important for lipidating LC3B, which we utilize as a marker for autophagy induction. This autophagosome then fuses with the lysosome, and we get the recycling of nutrients that's required to restore energy balance inside cells. And so one of the first things that we wanted to do is ask, in our CTR1 knockout mast memory fibroblasts, if we restore copper balance by putting in the human transporter or an empty vector, so these cells are about 50% reduced in copper transport, what happens to autophagy induction in the absence of amino acids? And so here we're looking at amino acid deprivation, and we utilize the inhibitor bathomycin. This prevents acidification of the lysosome. So you see buildup of these autophagosomes. And what we found is, in the context of the absence of the copper transporter CTR1, we see reduced lipidation of LC3B, which is this marker of autophagy. So autophagy is reduced, especially amino acid deprivation stimulated autophagy in the context of low copper availability. And this correlates with a reduction in the ability of ONC1 to phosphorylate its targets. So in the absence of amino acids, ONC1 is activated because TOR is inhibited and off of the lysosome surface. So you see increased phosphorylation of one of its downstream targets, ATG13. However, in the absence of the copper transporter CTR1, you don't see this increase in the phosphorylation of ATG13, suggesting that copper is critically important for both autophagy induction through this ONC1 dependent mechanism. And so here we were really quite interested in this phenotype, because we knew that if it was going to be a signaling initiating event, that maybe copper availability would be connected to these changes in amino acid abundance. And so here we're just taking our mouse embryonic fibroblasts. We're using a CF4 copper probe, which tells us how much copper is available in these cells. When there's a plentiful amounts of amino acids, we don't see major differences in the availability of copper. However, when we reduce amino acid availability, we start to see this increase in fluorescence, suggesting that there's an increase in copper levels inside these cells that correlate with autophagy induction. And so here we're able to see over time that there's an increase in copper availability, and that helps to sustain ONC1 activity. And so to transition to the cancer part of this, we wanted to identify a cancer type that was critically dependent upon autophagy for survival. And so it's been really nicely shown by Eileen White and Ali Kimmelman's groups that many of these ubiquitin-like conjugation enzyme, HTG5 or ATG7, are quite important in BRAF or KRAS-driven lung tumors. So if you knock out these proteins, you can see an increase in survival in the context of BRAF-driven tumorigenesis. And so we have previously characterized what happens in the context of an activated variant of BRAF in lung tumors. So if you cross these to CTR1 flox animals and look at survival of these animals, you're able to extend survival if you get rid of the copper transporter CTR1. So our next question was whether or not that was due to on-target inactivation of both of these signaling cascades. But first we looked at a fixed time point and monitored tumor burden in these mice. We found that there was about a 50% reduction in tumor burden in the absence of the copper transporter CTR1. This correlated with the reduction in the phosphorylation of ERK1 and 2. And importantly, some of these tumors still have MAP kinase pathway activation. And we think that that's due to the fact that there's only about a 50 to 60% reduction in copper availability in many of these tumors, but yet there's still a significant reduction in MAP kinase pathway activation. And so we went back to many of the tumor cells that, or the tumors that we had, and looked at autophagy levels by staining for LC3. We see that there's a significant reduction in LC3 positive vesicles in these CTR1 knockout cells, suggesting that both MAP kinase pathway activation and autophagy are reduced in these BRAF-driven lung adenocarcinomas. And so because we had both dual targeting of both of these signaling transduction pathways, we wanted to understand how much the reduction in tumor burden had to do with a reduction in autophagy levels in these tumors. And so we went to an assay that really allows you to understand the growth-like properties that are dependent upon nutrient depletion. And so here we would take our cells, get rid of amino acids, and look for the ability of these cells to survive that starvation. And the ability of those cells to survive the starvation was dependent upon autophagy induction. And so what we find is that in our CTR1 knockout cells, where we've replaced with the human copper transporter, we see that after starvation, these cells are able to survive, but in the absence of the copper transporter, these cells no longer survive. And this is recapitulated in the context of endogenous BRAF mutations in this lung adenocarcinoma cell line. So copper is really important for autophagy-induced survival in the absence of nutrients. And so finally, we wanted to connect this phenotype to ALK1's copper binding capability. And so we made mutations at these conserved residues that we had previously characterized to be important in MEK1 and 2 copper binding in ALK1 and 2, and then reduced its ability to bind to copper, and looked at autophagy induction in these cells. We found that as we reintroduced the wild type protein, that you could see increases in LC3/2 staining. However, that was diminished in the absence of in the presence of this copper binding mutant of ALK1, and that correlated with a reduction in the phosphorylation of ATG13. And so when we took these same cells and injected them into immunocompromised mice, we see that there's a reduction in tumor growth over time when those cells are expressing this ALK1 copper binding mutant. So the ability of ALK1 to bind to copper is critical in the context of both BRAF and KRAS-driven lung tumors, and this correlates with a reduction in autophagosome numbers. So the ability of ALK1 to turn on that autophagosome or autophagy program is reduced in the if it can no longer bind to copper. And so finally, we were really interested in these genetic observations that MAP kinase pathway activation and ALK1 and 2 induced autophagy that are critical for BRAF-driven tumorigenesis could potentially be leveraged in the context of KRAS and BRAF mutant lung adenocarcinomas. And this was really driven by the fact that many labs have recently showed that on-target inhibition of the MAP kinase pathway or BRAF or KRAS themselves leads to paradoxical activation of autophagy in tumors. So that is a pro-survival pathway that gets turned on. However, and that requires targeting of the MAP kinase pathway and autophagy in these tumors. However, in the absence or the presence of our copper chelators, potentially, we would be targeting both nodes of this signaling pathway. And so indeed, if we take these BRAF mutant lung adenocarcinoma cell lines, and we treat with the MAP kinase inhibitors trametinib or vemurafenib, we see that there's an increase in LC3/2 staining in these cells, suggesting that autophagy has been induced, and this correlates with increases in phosphorylation of Becklin and ATG13, which are downstream of ALK1 and 2. However, if we treat with a copper chelator TTM, we see that that's reduced, suggesting that you can reduce autophagy when you utilize the copper chelator, and that's due to on-target loss of phosphorylation of ATG13 and Becklin-1. And so when we take these same cells and do this starvation-induced survival-based experiment, what we find is that those tumor cells that have been treated with the MAP kinase inhibitors trametinib or vemurafenib, don't really succumb to that survival-induced death, or that starvation-induced death, because they've induced autophagy in these tumor cells. However, they're not able to induce that autophagy in the copper chelator treated context. And so finally, what we've really showed is in the context of oncogenic BRAF or KRAS-driven tumorigenesis, specifically in the lung, that copper is required for ULK1 and 2 driven autophagy. And most of this work was done by 2 graduate students in my lab, Tiffany Zhang and Caroline Davis, 2 postdoctoral fellows, Jessica Pasimo and Xing Gu. And we were aided in a lot of this work by my colleague, David Felser and Andrea Gudial and Michelle Cicchini in his lab. And so what I hope that I've really told you about today is that we've known for many years that copper is critically important in the context of energy maintenance and metabolism and the synthesis of biocompounds like DPH and lysyl oxidase. However, we've started to scratch the surface of copper's ability to function as a signaling molecule and be important in these cellular processes that are really important in the context of cancer biology and other diseases. And so with that, I'd like to thank the people that are listed on this slide that have really been helpful in doing much of this work, and the many members of the Brady bunch over the last 7 years and our funding sources. And I'd be happy to take any additional questions. Yeah.
So I was just wondering, so obviously this is preventing the rewiring that's happening with the BRAF inhibitor. Do you think that if kept long enough, there will be more rewiring that happens with?
Yeah, I definitely think that that's true. So we do know, at least in the context of melanoma, if there are other pathways that are activated, PI3 kinase and others that we don't see as being copper dependent, that you can get resistance to the copper chelator therapy, suggesting that you need another combination therapy. We haven't been able to ourselves create a resistant pool of cells if we treat with the copper chelator, but that doesn't mean that it can't be done. Yeah.
That's for like all of these important kinases that potentially.
Yeah. So that's a really important question that we're trying to unravel now, which is what is the hierarchy of need between these pathways? What we at least know is that, for example, the copper regulated autophagy phenotype isn't really in play unless nutrients are scarce, right? And so you're not seeing changes in that pathway in any cell that has reduced copper availability. Now, for the other pathways, I think it's going to be about how they get their copper and what the affinity actually is for copper. And so we're now trying to do some additional studies to understand those biochemical properties.
These were activated in response to mitogens, because I'm thinking like if it's like way higher in a apparently activated cell, then maybe will be better tolerated.
Yeah, sure. Yeah, absolutely. There are some tumor types where you see overexpression of CTR1 and some of these other transporters that may lead to a therapeutic window for sure. Right. Yeah.
Oh,
Okay, my question is about your CCS. Is there any thought, you mentioned I don't know if it's a specific peptide, is there any thought to like a peptide mimetic, like a BH3 SMAD type of, is the peptide specific enough to like copper binding?
Yeah, that's a great question. Yeah, I think you could probably make a peptide that would fit there. Definitely, based on the small molecule modeling, I think that's definitely a possibility. Yeah.
To piggyback on her question about the dosing, this
Yeah.
is a neat idea about like the pulsing ERK signaling, and where do you think yours fits in with getting like the cells getting addicted in resistance to like the ERK inhibitors or something?
Yeah.
Anywhere that you see you dose at the same time then as the inhibitor, do you let them kind of develop resistance and then come in with your copper?
Yeah, so we've started to look at this a little bit by using the ERK sensors. And there it got really interesting. So we were using this new OptoSauce, a construct that's been characterized by my colleague Lucas Bagage. And so there we're able to activate the MAP kinase pathway by different pulses of light for long periods of time and all those things. And so then when we treat with a copper chelator, what we found is that only subsets of the population are actually responding. And and they've gotten the same exact amount of light, but we don't know why some of the cells respond to the chelator and some of them don't. It could be cell cycle stage. It could be how much the pathway has been activated. So I think there are some additional questions that will be helpful to understand so that we know how to use the chelator the best. Yeah, I think you're exactly right.
So the question on mine, is there any way to track subcellular copper concentrations?
Yeah, absolutely. So we utilize these biosensors or these fluorescent sensors that were developed by my colleague, Chris Chang at UC Berkeley. The ones that I was showing here were mainly copper 1 selective, but we recently just published a paper on one that's copper 2 selective, and there we're able to track the labile pools of these of oxidation selective amounts of copper that are available. Yeah. And they're about in the picomolar range. Yeah.
So I'm wondering about the sort of the push and pull of whatever copper is doing for mitochondrial homeostasis and the chelation and also the disruption of even kinase path.
Yeah. So one of the things that we know that I think is perplexing to the field is that these CTR1 knockout cells that we utilize for a lot of our studies don't have a defect in mitochondrial copper. So for whatever reason, it's prioritized. Yeah, well,
Just a concentration.
I think it's a concentration. I think honestly, for the copper that does come into the cell, the cell has rewired to make sure that it maintains mitochondrial copper pools. We've now started to look at what happens in our cells if we only specifically manipulate the mitochondrial pool. Interestingly, we see that there are still defects in the MAP kinase pathway. So there's some connection between the mitochondrial pool and these cytosolic signaling pathways that we're trying to understand now. We think it's connected to mitochondrial metabolites that are generated there and their effects on the pathways. But
Solute carrier more efficient for taking whatever copper is available?
yeah, that would be a really good question. I don't think none of the work that's out there to date would really address that. Yeah, it could be. It could be.
One more quick question.
Yeah.
Is there in the CTR1 knockout mouse? Do they live?
No, it's lethal. Yeah, it's a great question. No, they die at like 9.5 or something like that. It's
They
lethal.
get through gas relation.
They do.
Relation.
Right.
They make their own press.
Oh, that's a great question. I'd have to look back. I'm not sure.
Great.
Yeah. I'm happy to look. Yeah, that would be. Yeah, it's good to know.
I'm wondering whether you can comment on interaction crosstalk between AMPK and M4 in this case. AMPK seems to activate ORF1, suggesting that that's not something we should do to activate AMPK. Because M4, in your case, it's a de-repression of that leads to the MEK activation. And ACAR seems to also inhibit that.
Oh, yeah.
Seems to be good for the treatment of cancer, but then at the same time, MPK activated.
Yes. So we have looked at AMPK activation in our cells. It is activated as expected because the ATP levels are lower, so we do see that. We haven't necessarily looked at the interplay with the MAP kinase pathway though. Yeah, that's something that we haven't done. We don't see changes of the ULK1 site that's put on by AMPK though, at least in the CTR1 knockouts. Yeah, we don't see changes in it. We do, and we don't see changes either in the TOR site either, right? Because neither one of those kinases are directly regulated. But we do see changes in phosphorylation of ACC, for example, or phosphorylation of AMPK itself. Yeah.
So that's like that way for one.
after the talk. She will have lunch meeting with graduate students. There, but
Yeah.
And.
Oh.